Dear students, welcome to another lecture on bioinformatics, and today's topic is on DNA sequence analysis and gene hunting. The major objectives of today's lecture are to acquaint the students about the need of DNA analysis, to make them aware about the gene structure and DNA sequences, to give them an idea about gene hunting and its importance, to tell them about the positional cloning and RNA transcript analysis, and to give them a brief account about the hierarchy of genomic information. Dear students, let's begin with the introduction. The extraction, sorting and analysis of sequence information about genes, genomes and proteins are one of the major aspects of bioinformatics. Contributions from the field of biology and chemistry have facilitated an increase in the speed of sequencing of genes and polypeptides. With the advent of cloning technology, it has become easier to insert foreign DNA sequences into many systems. A rapid mass production of particular DNA sequences, a necessary condition to sequence determination, has also become possible through the use of modern tools and techniques. Oligonucleotide synthesis technology has allowed researchers with the ability to construct short fragments of DNA with known sequences. These oligonucleotides could then be used in probing vast libraries of cDNA to extract genes containing that sequences. Alternatively, these DNA fragments could also be used in polymerase chain reaction to amplify existing sequences or to modify these sequences. Two common goals in sequence analysis are to identify sequences that encode proteins which determine all cellular metabolisms and to discover sequences that regulate the expression of genes or other cellular processes. The information is obtained by sequencing of genes. For this, they have to be isolated and cloned in the laboratory. So, it becomes essential to understand the biological background of how sequences are obtained. After many successful sequencing projects, huge number of sequences are made available and the number of sequences are still growing rapidly. Due to the increasing number of sequences, the quality of statistical analysis needs to be improved. While the development of new tools for identification of biological function from the sequence patterns allows faster detection and cloning of new genes. Dear students, let's now discuss about why to analyze DNA. Sequence analysis is the process of trying to find out something about a nucleotide or amino acid sequence, employing in silico biological techniques. The in silico techniques come into picture when you may have sequenced a gene by yourself and then you wish to learn what these long string of letters, that is the nucleotide bases, represent and actually code for. You may want to confirm that you have indeed cloned a gene successfully, or you might want to learn about a sequence of DNA that you know absolutely nothing about. You may even want to know if a fly has a similar protein to a human one. In these and many other situations, you may use of sequence analysis. Besides, there are numbers of applications of sequence analysis in the field of biology and allied sciences. Due to rapid progress in algorithm development in computational biology, large number of softwares are freely available that can compare unknown sequences to all the sequences in the database. Sequences that are similar to the query sequence will be identified. There are large numbers of databases that contain genetic information and are publicly available. Currently, many of the international scientific journals require these sequences to be submitted in the publicly available database before a newly discovered sequence can be published. Large numbers of sequences have been checked and published, often annotated and cross-referenced. Each record is curated and maintained in one of the many different databases accessible over the internet. Each database has its merits and demerits, and you will learn to choose the most appropriate database for your desired search. There are many advantages of performing sequence analysis, for instance, to predict which sequence are coding are those that present with a graphical three-dimensional image of the macromolecule. The initial facts carried out on a new sequence is a map of repeat locations to find out the likely occurrence of regulatory and protein coding regions. Strong evidence of exons can be predicted by doing sequence similarity search, and this might also reveal the functional sites. data box, splice junctions, etc. on the DNA. There are some more tools which determines the restriction mapping here by using a small piece of DNA sequence. The recognition sites by the restriction enzymes can be found out, finding the ORFs, checking codon preferences. Organisms tend to use certain codons more than others. Finding polymerase third promoters, finding PCR primers, melting temperature of the sequence, and many more can be found out from any new DNA sequence. Once all these information are obtained, they can be gathered together and a complete picture can be drawn and analyzed for further studies. All protein sequences, whether sequenced or obtained through translation from ORF in a nucleotide sequence, contains valuable information on its structure and function. Unfortunately, the raw sequence data of protein or nucleic acid outnumbers the structural data. This is mainly due to the amount of time and cycles involved in determining structural features. This forces us to adapt a predictive methodology for finding new structures computationally using the previous structural data. Proteins are made up of 20 chemically different amino acids that allows for greater diversity in structures and function. Due to each residue different physical property, it has been found that the domain is adopted different structures with different amino acid combinations that makes it rather complicated to analyze compared to nucleotides. The initial sequence analysis carried on any protein sequence are composition analysis, molecular weight search, isoelectric point calculation, peptide mapping by exposure to proteases or chemical agents, hydrophobicity and hydrophilicity of the sequence, secondary structure determination, fold prediction, transmembrane region prediction, coil structure prediction, signal peptide prediction, motif prediction, non-globular region prediction, tertiary region prediction, and so on. Once we obtain a new sequence, or if we have a sequence of interest, we will see how we go. The stepwise sequence analysis can be performed using tools that are available on the web. Step first, to obtain sequence of interest. If you already have a sequence of interest, you can proceed to step second. In step second, identify ORFs and translate them into protein. In step third, find similar sequences in the database. In step 4, do an alignment of sequence or sequences. In step 5, look for multiple sequence alignment. In step 6, look for the presence of specific patterns in your protein. In step 7, find similar sequences in other species. In step 8, determine the putative structure of your protein. In step 9, obtain function of query proteins. In step 10, input your sequence into an alert server. Dear students, let's now talk about the gene structure and DNA sequences. A gene is a sequence of chromosomal DNA that's required for the production of a functional product, that's a polypeptide or a functional RNA molecule. A gene includes not only the actual coding sequences but also adjacent nucleotide sequences required for the proper expression of genes. There are certain key features of eukaryotic genes that needs to be assimilated in order to understand the impact of gene structure on sequence analysis. These include, for example, introns, exons, and coding sequences, untranslated regions, and so on. Prokaryotic genes generally lack introns and consequently have a somewhat simpler structure. DNA sequence database typically contain genomic sequence data, which includes information at the level of the untranslated sequence, introns and exons, mRNA, cDNA, and translations. Thus, it can be seen that so-called DNA databases in fact contain a variety of different types of data that cannot all be treated alike. These refinements affect the manner in which search results must be interpreted. For example, searching cDNA for intron-exon boundaries, which only occur in genomic DNA, would be rather futile exercise. Dear students, let's now talk about the untranslated regions. Untranslated regions, simply UTRs, occur in both DNA and RNA. They are portions of the sequence flanking the coding sequence that are not translated into protein. Untranslated sequence, particularly at the 3' end, is highly specific both to the gene and to the species from which the sequence is derived. Dear students, let's now talk about the conceptual translation. Given a piece of DNA sequence and knowing the genetic code, it's possible to translate the DNA into protein by looking up successive codons in a genetic code table. This is termed the conceptual translation. It's important to distinguish between sequences for which the translation has some biochemical support and those that are simply derived theoretically or computationally. The use of the term conceptual indicates that the specified translation is only theoretical and carries no experimental validation. Dear students, let's now talk about the features of DNA sequence analysis. Determination of the complete genomic DNA sequence of an organism allows attempts to be made to identify all of an organism's genes and therefore define its genotype. Special experimental techniques have been devised to carry out the difficult task of manipulating and characterizing large numbers of genes and large amounts of DNA. One such approach to genome sequencing is first to generate high-resolution genetic and physical maps of the genome to define segments of increasing resolution and then to sequence these segments in an orderly manner. Another approach, the direct shotgun approach, is to break up the genome into random overlapping fragments, then to sequence the fragments and assemble the sequences using computer algorithmic programs. Analysis of genomic sequences reveals that each organism has an array of genes required for basic metabolic processes and genes whose products determine the specialized function of the organism. Complete genome sequencing therefore provides a knowledge base on which to build information about gene and protein expression, but is not sufficient on its own to define the entire protein component of the organism. Dear students, let's now talk about the gene hunting. In human genetics, gene hunting is the process of finding genes that contribute to individual differences in phenotypes. The phenotypes may be diseases and disorders or part of normal variation. The process of gene hunting begins with a well-defined phenotype. The term well-defined is relative to what is already known about a phenotype. For instance, many of those involved in the research and treatment of psychopathology suspect that our current view of schizophrenia really encompasses a number of different but perhaps related disorders. We simply have not been clever enough to distinguish one disorder from another based on phenotypic symptoms and signs. The fact that schizophrenia is still somewhat ill-defined in this way does not dissuade research from gene hunting for schizophrenia. In contrast, many of these researchers hope that the discovery of genes for schizophrenia may be a rosette stone that can assist in solving the problem of heterogeneity. Many human phenotypes relevant to social science are continuous or quantitative traits. A continuous trait is a phenotype that everyone possesses, but we have different amounts of it. Height is a classic example. It is not that some people have height and others lack it. We all have height, but we are not all equally tall. Gene hunting can be done, in fact is currently being done for continuous traits. A gene contributing to a continuous or quantitative trait has become known as a quantitative trait locus or QTL. In recent years, substantial financial resources have been expended in the search for new genes that may be linked to a particular type of disease. The aim is to develop new therapies with which to combat a wide variety of preventive disorders such as breast cancer, asthma, and neurodegenerative diseases, and many more. There are two main strategies for discovering proteins that may represent suitable molecular targets, whether for small molecule drug discovery or for gene therapy. Dear students, let's now talk about the positional cloning. The first approach for discovering disease-related genes is the technique of positional cloning. Here the chromosome linked to the disease in question is established by analyzing a population of subjects, some of whom exhibit the disease. Once a link to the chromosomal region has been established, a large part of the chromosome in the vicinity of this region, known as a locus, is sequenced, yielding several megabases of DNA. Such a locus contains many tens of individual genes, only one of which is likely to be involved in some way in the disease process. Sequence searching and gene prediction techniques can be used to increase the efficiency of gene identification in the locus, but ultimately several genes will need to be expressed and further experimentation or validation will be required to confirm which gene is actually involved in the disease. Although genes discovered in this way can be very illuminating from an academic point of view, they do not necessarily represent good drug targets or points of therapeutic intervention. The whole process is lengthy, time-consuming, and labor-intensive. Dear students, let's now discuss about the RNA transcript analysis. The alternative approach to gene discovery, requiring much less sequencing effort and relying more heavily on the powerful search capabilities of current computer systems, examines the genes that are actually expressed in healthy and diseased tissue. This allows a comparison to be performed between the two states and a process of reasoning applied to arrive at a potential drug target in a more direct way. This process analyzes the mRNAs, which are used by the cellular machinery as a template for the construction of the proteins themselves. Dear students, let's now talk about the hierarchy of genomic information. The human genome is complex, consisting of about 3 billion base pairs of DNA. Yet only 3% of the DNA is coding sequence, that is, that part of the genome that's transcribed and translated into protein. The rest of the genome consists of areas necessary for compact storage of the chromosomes, replication, at cell division, the control of transcription, and so on. A large part of the work of sequence analysis is centered on analyzing the products of the transcription, translation machinery of the cell, that is, protein sequences and structures, where the latter are available. Recently, much industrial emphasis has been placed on the study of mRNA. This is partly because a conceptual translation into protein sequence can be generated readily, but the main reason is that mRNA molecules represent the part of the genome that's expressed in a particular cell type at a specific stage in its development. Thus, in simple terms, we have three levels of genomic information. First, the chromosomal genome, or simply the genome, the genetic information common to every cell in the organism. Second, the expressed genome, or transcriptome, the part of the genome that's expressed in a cell at a specific stage in its development. And third, the proteome, the protein molecules that interact to give the cell its individual character. These are the three basic levels, but others could be envisaged. Example, the metabolome. For each level, different analytical tools and interpretive skills are required. Tools appropriate for the discovery of intron-exon boundaries in genomic DNA may be interpreted in misleading ways when applied to mRNA data, sequence visualization at the DNA, RNA, and protein levels, and distinct analytical techniques sharing some common threads. But again, correct interpretation of the results is of paramount importance. The skill lies both in choosing the right tool for the job at hand and in understanding the intricacies of the various analysis outputs. This is an end of today's topic. Hope you have enjoyed and understood it well. Thank you.